Hello, everyone. I'm Himel. Um, I am a senior scientist at Mark. I recently joined Mark from Huttenhauer Lab, which is at the Harvard School of Public Health, where I did my postdoctoral fellowship in microbiome multiomics data analysis around it. So I'm going to talk about, and I'm very excited to talk about my new tool, Tweedieverse, which is a general purpose differential analysis tool for microbiome multiomics data. And I must say that I'm looking forward to interacting with everyone at the BBCN conference, both regarding any questions or clarification regarding the talk, as well as for general chit chat regarding bioinformatics and microbiome data. So my talk is separated into 5 parts in total. So the first part will be on what is the current state of microbiome multiomics differential analysis. And I'll move on towards the motivation and challenges. That was the main reason that we wanted to build this tool. The tool, as you can guess from the title, is Tweedieverse. So how we have built Tweedieverse and how we have validated it, that will be the third part of the talk. And then I will show an example apart from some of the synthetic benchmarking as part of the validation we did. Also, a real data application using the integrated human microbiome project that was recently completed in 2019. Yeah, the final section, I think that's my most favorite section because apart from the concluding remarks and next steps, I would also show you how to use this tool for your own purpose. This talk, even though it's human microbiome focused, there is a component that I would like to address, that if somebody have data set, microbiome data from different environments, how Tweedieverse, this tool can be helpful, not only for the purpose of differential analysis, but also for the purpose of benchmarking. So with that, I would like to go to the first part of the talk, which is the background and motivation. And throughout this talk, I want to share this logo that we have created for this tool. As you can see from the logo, Tweedieverse tries to simplify the process of differential analysis, as you will hear from me in a bit. That's what is described in the logo, that let Tweedieverse handle it. And that will be also the take-home message possibly that I would like you to take from this talk. The first part, I should, at the very beginning, I should clarify that I'm going to also use the term multi-omics a lot. Again, multi-omics has different meanings. In general, usually integrated omics, which is shown in this figure, that from a single omics you get one set of interpretation of your data. But when you have multiple omics, you usually get a more comprehensive viewpoint of the biological phenomena that you are interested in investigating. But in the sense of, at least for the purpose of this talk, multi-omics can mean taxonomic and functional profiles from the same metagenomic samples. That's also, in my mind, multi-omics, because you do have two different layers of data, even though they are coming from possibly same biological specimen. On the other hand, you can also have multi-omics, for example, metabolites, metabolomics measured in stool and plasma, as well as metagenomics, taxonomic profiling, again measured from stool. Those are also great examples of multi-omics, where you have two data layers coming from possibly two disparate sources. So I'm going to cover both types of multi-omics throughout this talk. Yeah, I think I just wanted to emphasize that in general, we are going towards multi-omics nowadays, with the hope that with more data, we are possibly going to understand the underlying biology better. So sum is greater than parts. With that, I think with this viewpoint, I think it will be clear why some of the methods, for example, differential analysis, becomes more and more important when analyzing multiple omics together. As the data increases, I think the responsibility of analyzing those data also increases. I would like to start with an example microbiome multi-omics data. And as I said, it's pretty much generally applicable to any multi-omics data set with or without microbiome. But for the sake of this talk, so this is an example of a multi-omics data or multi-omics study where the measurements are coming from various sources, typically same biological specimens, but the data layers that we were interested in are metagenomics, metatranscriptomics, metaproteomics, and metabolomics. I think there are several ways to analyze multi-omics data, but again, for the sake of this talk and also for the purpose of introducing Tweedieverse, I think one of the goals that usually in this type of multi-omics studies people ask is, how do I identify clinically actionable biomarkers from integrated multi-omics profiles, so that there is a possibility of doing better science or better therapeutic development, especially in a context of drug development, for example. At the core of the problem, I'm going to simplify to saying that differential analysis is a bulk of that, because we are interested in finding biomarkers, which is again a general term, based on the microbiome for the purpose of further investigation. So if we can identify a feature from anywhere from the microbiome multi-omics that is differentially abundant or expressed, I think that forms the basis of any follow-up investigation. So I'll summarize by saying at the core of the problem is differential analysis. And I know that we are possibly all familiar with the term differential analysis, but what I have done here is I'm reintroducing the differential analysis by combining differential abundance and differential expression. So differential abundance is usually more popular for DNA-based measurements, and similarly, differential expression is more popular for RNA-based measurements. Since in the microbiome community, differential abundance analysis is possibly the most common form of analysis as compared to, for example, RNA-seq literature from the host side, where differential expression analysis is the most common form of analysis, especially when you have a two-group scenario where cases and controls, which is simplest scenario. But I'm redefining it, just saying in my particular case, since I am in a more general setup of multi-omics, I'm defining it as the differential analysis refers to the class of problems where one tries to identify features anywhere on the omics spectrum. That's the important part, that are either over or under abundant or expressed in some condition related to a reference condition. Again, with this definition also, I do need to pinpoint certain details about what I exactly mean by differential analysis, because in the microbiome community, there are several ways to do differential analysis. One can do some sort of omnibus testing based on alpha diversity or beta diversity, but there is a class of method that I am interested in are called per feature differential analysis. I think arguably with the per feature approach, the output that you get are more granular, in the sense that you are able to tell which particular features are differentially abundant or expressed, as compared to saying some measure of the whole data set or the whole community in terms of, for example, diversity. So the end goal is you have layers from, or data set or layers from, for example, DNA, RNA, proteins, metabolites, and so on. I think the end goal is to see or be able to tell what are the DNA features, what are the RNA features, and what are the other biochemical features that are differentially abundant or expressed with respect to the disease. So currently there are not many tools that are capable of doing multi-omics association, in the sense that they are either optimized for DNA type measurement, or they are optimized for RNA, or they are optimized for other type of data, but not all of them. So that would be one part of the talk that I'm going to emphasize, that with the differential analysis, we would like to see a picture like this that we are seeing here. And because this is a per feature approach, mostly in a statistical term, it's a univariate. That means we are analyzing one feature at a time. And here the notation is y is my feature of interest, the typical regression setting y tilde x, that we are trying to regress y on the metadata x. And in the simplest scenario, x is just the binary variable that you're interested in, but there is a possibility, or there is opportunity to also include multiple covariates as well as repeated measures. There are several issues with type, because usually this is the most popular form of differential analysis, but there are several statistical challenges that remain despite the fact that it has been a well-studied area. So typically, I think just to begin, there are many tests, because depending on what data type we are talking about, we can have 100 to million features that we need to test. So that means high dimensionality is intrinsic to this problem. So there will always be many tests, which means there is a need for multiplicity correction, FDR, false discovery rate correction as well, which is not really always straightforward. And also, because of the noisy and non-Gaussian or non-normal distribution of the features, there are several analytical challenges that you cannot take an off-the-shelf tool that has been developed for more normal-like features. You can possibly not apply those to this type of data sets. And also, most of the newer, I think, measurements are counts. So it's recommended that count models rather than continuous models be applied on them, so that the intrinsic property of the data remain intact. So with that, there comes the challenge since it's most of the measurements are count based, there comes the challenges or extra challenges of mean variance dependency, zero inflation, repeated measures, and so on, as the design of these studies go more complex. So at the end, I think the last point that I want to make is that well-calibrated p-values, in the sense that if you really don't have any signal and you draw a simple histogram, I think the expectation is that it's uniformly distributed if there is no signal in the data. So if that's not the case, that means the p-values are not well calibrated. So well-calibrated p-values as well as false discovery control has not been straightforward in some of the microbiome data because of the challenges that they have, which I'm going to explain a little bit later in a separate slide. To summarize, several methods exist, but without a clear consensus and no gold standard. Now, I cannot make this statement for multi-omics or a single omics or all the single omics, I think I should say, but definitely this is the case for most of the single omics literature. There are certain data types that are possibly more saturated, in the sense that there are gold standards that exist, but most of the time I think several methods exist, but there is no gold standard, especially with the microbiome multiomics. To belabor this point a little bit more, usually when a data set is collected after a study is conducted, the first question a biologist wants to ask is, I want to do differential analysis of my multiomics profiles, and I'm assuming that there are more than one modalities, and you can imagine that the modalities can become, if with the number of modalities increasing, this question becomes difficult. I think that typically the current workflow goes in 3 branches. Here on the left-hand side branch, I think you have to consider what is the best way to normalize my data, because I know that there is a sequencing depth variation across my samples. So I cannot really compare the samples. So I have to somehow normalize the data. I can do very simple rarefaction or some TSS, total sum scaling normalization, that are popular, but I'm not sure whether I can do that for all my omics layers that I have collected. Um, and usually the answer, if you ask this question, what is the best way to normalize my data? Most of the cases, the answer is choose your favorite one, because there is really no gold standard when it comes to normalization method for different data modalities. Now, once you have settled on one normalization or transformation scheme, I think the second bullet point that you need to address is, if my data is zero inflated, is it non-zero inflated? And you have to ask also the question, is it count, and following the normalization and transformation, is it continuous? So depending on all these four combinations, again, there is no one answer, or at least a consensus answer. So you have to always go back and see what is my data type, and depending on that, you have to choose your favorite statistical model. And this is really with respect to one binary metadata. That means you have a very simple case control type of scenario, or disease healthy type scenario. But if you have more metadata or more covariates to adjust for, which is typically the case nowadays in population-based microbiome studies, and also you do, if you have repeated measures, because that's also another aspect, longitudinal aspect, which is becoming more and more common nowadays, then also this becomes much more complicated. Especially some of the existing tools that people are using for many days, once the design gets a bit complicated, it's very difficult to apply those same tools for this purpose. So I think the point here is, as a practitioner, I have to answer these three questions: how to properly normalize my data, to make sure the between subject comparisons are meaningful. Once I have done that, I need to consider what statistical model to consider. And this is a big question, because at the end, my p-values, if they are not well calibrated, my decision can be completely wrong, and that might impact the whole study objective. And depending on my complexity of the experimental design, I have to further decide whether the model that I have chosen is also able to handle repeated measures, and also adjust for covariates if I want to. And on top of that, with this difficult workflow, there are currently more than 100 differential abundant tools. This is partly because not all of them are designed for microbiome data, but people have taken tools from other fields. For example, host RNA-seq, dSeq2-EDGER. These are two tools that are popular in the bulk RNA-seq literature. Those have been applied to the microbiome data, as well as Lima, for example, which is one of the first tools for differential expression analysis. So in summary, there are more than 100 tools, and as a practitioner, especially as a newbie, it's very difficult to choose one method over the other, and it becomes complicated when there are multiple omics layers are present. This is also, there is another reason why this has become complicated, especially for microbiome multi-omics data. So if we closely look at what are the properties and nuances of microbiome multi-omics data, we usually think of m number of features and possibly n samples, where m is usually much greater than n, especially in a small experiment setup, and also even in a population setup, usually m is very, very large. But this is not surprising. This is possibly all genomics data, and all genomics data are exactly like this, high dimensional in nature. So there are certain qualities that microbiome data share with other modalities or other omics data. But in microbiome, I think one interesting part is that there is a hierarchical nature in the features because of the phylogenetic relationships. But depending on what data layer we are talking about, as it if we are talking about taxa, then it's around 100 to 1,000. I'm mostly talking about human microbiome at this point, but this is roughly than I estimate and this has possibly changed. This is an old slide as well. But I think the point is going from taxa to genes usually increases the number of features as compared to, for example, functional modules, which are summarized features at the pathway level, are possibly in between. So in terms of how the data looks like in the columns, so in here the columns are usually samples, but in microbiome data can be pretty diverse in that sense. It could be diagnosis information, or it could be environment type. Mostly these are subject or sample specific metadata that we want to connect or want to link with our microbiome data. As I said, since this high dimensional nature of microbiome data is similar to other existing omics layers, there are some common challenges or common issues that are not unique to or specific to microbiome data. I have listed all of those there in the left-hand side, but I think the point is that you do need to, I think going back to my previous slide, you do need to take care of multiple testing, library size differences across samples, as well as some of the count-based characteristics such as over-dispersion, non-normality, mean variance dependency. And then with the design becoming more and more complicated, that more and more metadata, you do also need to take care of some of the non-independence of the metadata that you are interested in testing. But for microbiome data in particular, we have more challenges because the measurements that we get are compositional in nature. And also there is a zero inflation, that there are so many zeros. This is particularly interesting because zeros can occur as a result of two reasons. One is technical, that a certain species get undetected due to the limit of sequencing depth, but also it could be true biological zero. That means it could be a rare species, but we don't know usually from the measurement type. So that creates the problem of zero inflation that we need to somehow tackle in the analysis that we do on this data. And as a result of all these challenges, FDR control, which I'm going to talk a lot today, is actually not guaranteed. In the previous studies, FDR control was taken for granted, but later on, people found that FDR control can be a big issue if some of these challenges are not well taken care of during the analysis. I want to switch gear a little bit just to carry on this motivation about how many tools are available, how is it difficult for a practitioner, especially a newbie, to think about what is the best way to analyze the data. In the single cell host RNA-seq literature, single cell has become popular in the last few years. So one of the reasons I wanted to take the example of single cell is that single cell data sets are very similar to microbiome data. One thing that has happened over the last few years is that people have taken a tool that was developed for microbiome and applied it to single cell differential expression and vice versa. This is because I think once you get the data metrics that I showed in the previous slide, that you have m number of features and n samples, and if you have these statistical properties like zero inflation and some other idiosyncrasies, I think that becomes statistically very similar problem. So that is the reason when the data sets are very similar, even though they have different biological interpretation, methods can be applied across omics. So that has happened definitely with the host single cell RNA-seq and microbiome. So one particular thing that I wanted to talk about from the single cell literature is that single cell literature, in the single cell literature, it was assumed, or it was taken as the knowledge that usually single cell data is zero inflated, which was based on a different technology. I don't want to go too much details there, but recently a new technology, droplet scRNA-seq, has come, and there was evidence from this paper that I'm showing here that's showing that this new technology-based single cell RNA-seq is actually not zero inflated. And to confuse the readers a little bit more, because from the point of view of all the methods development that has happened over the years, assuming that this data is zero inflated, it was a little bit of bad news for at least for those methodologies. And then there was this follow-up paper that came out, which is just showing that the new technology, droplet SCRN6, actually that is not the reason for zero inflation. Zero inflation is mostly related to what you measure. So there is a thing called UMI and non-UMI. I'm again not going into details there, but another paper showing up that it's not really the technology, which was the previous technology was called SmartSeq. The newer technology is called DropSeq. So it's not the DropSeq versus SmartSeq that you can really say the reason for zero inflation or not. This is actually what you are measuring. So if you are measuring UMI, it's not zero inflated. If it is not UMI, then it's zero inflated. So that is actually the consensus currently. So, but when I first saw that, I felt that if I'm a newbie in this field, it's a long-standing confusion, and it's not something that everyone agrees, but this is the most agreed upon definition so far. So the point here is whether the data is zero inflated or not, even that has generated a lot of confusion over the years. So if I want to again jump into the microbiome literature now, if we take a look at what are the different methods that have been developed in the microbiome world for the purpose of differential abundance. Obviously, when the microbiome shotgun sequencing as well as 16S technologies came about, I think around somewhere I think 10 years ago or more than that. I think there was obviously lack of methods or lack of comprehensive benchmarking, which is totally reasonable. But starting with the human microbiome project that completed in 2012, there have been several developments over the years. Again, I cannot go too much into details of each of these methods, but they try to solve at least one of the aspects that is not solved by previous methods. And you will see that in this list of methods, there are methods also from other omics data analysis. For example, bulk RNA-seq, you will see DSeq to EDGER, as well as Lima from the microarray. But there are also microbiome specific methods that try to capture at least one aspect, for example, repeated measures or zero inflation or something else that has not been addressed by other methods. So this is a great news, right? Because from the point of view of methods, that we have so many methods available, and we have a very good, very robust set of analytical methods depending on the design we can apply. But what happened from 2017 onwards, that there have been independent evaluations that are showing that most of these methods, at least some of these methods, have high false positive. And again, this is an old slide. I have found myself more than 20 independent evidence. I'm sure that number has increased by now. So what is the current scenario then? So in 2021, we are still possibly saying the same thing that we started with in 2012, that lack of gold standard methods as well as lack of comprehensive benchmarking. So some of this has been addressed in some recent papers, but that was also the motivation for the method that we are going to talk about. And this is my summary of what is the current state of differential analysis tools from a practitioner's perspective. There are obviously too many choices and too many options, which can be overwhelming. And also there are too much confusion, especially regarding what is the best way to analyze the data. As I have shown the example of single cell, I think it's not well agreed upon whether the data is zero inflated or not. In the microbiome also, there are similar arguments about compositionality, for example, whether you should be using a compositional approach versus a non-compositional, and there is not a clear definition as of yet. So my one-line summary, that what I've just said so far, is that there are several methods that exist, but without a clear consensus and no gold standard. So the second part that I'm going to talk about is, then given this situation with the differential analysis of microbiome multi-omics, what are things that we can do right and do a little bit better? So going back to the flow chart that we saw before, I think one of the questions that I wanted to ask, how do we resolve this long-standing interoperability problem across platforms and omics profiles? So every time a new platform becomes popular or becomes mainstream, do we really need to go back and develop a new method? Or can we use the existing infrastructure in an intelligent way so that there is a way to unify the complex workflow in a single estimation umbrella? So that's the motivation that I wanted to take. And again, Tweedieverse tries to solve some of the problems by not making the job difficult for the end user. So we will see that in a bit. How does that translate? I want to also quickly mention that this type of efforts where you try to unify some of the existing methods or existing theory is not uncommon. So here is an example of non-parametric data science. I don't work in this area, but this is just an I found this interesting that there is a blog post written by one of the pioneer of non-parametric data science that it seems like this is also the case in that field, that there is a dire need to put some order into the current inventory of algorithms that are mushrooming at a staggering rate. So I connected with this statement very well, especially when it comes to microbiome data analysis. Like there are so many tools being developed without really connecting them or making them talk to each other, or in the sense of how do we unify or combine all these methods to understand the biology better. This is really the second point. It's where it says that consistent and unified framework, so that you can reveal the interconnectedness among different branches, in this case different omics layers, I think is the possibly the end goal of what we are trying to do using a unified framework. But as I mean, possibly it's clear that we are really trying to solve a difficult problem here. We are talking about trying to come up with a method that works for most of the data types. So we are really saying differential expression and abundance pattern. So we know that biologically there is a differential patterns across different omics layers, depending on what data type we are talking about, what environment we are talking about, and also whether it's a healthy versus disease. So there are so many things going on. So we do want to capture that. I think possibly that's what we're trying to say. And then apart from that, I mean, apart from the overall pattern, there on a per feature basis, there are differential amount of sparsity, I will say, for example, zeros or zero inflation. These are also, usually they differ across environments or across data types. So there are two things going on. One is at the broad level, there is a difference across different modalities. And then at the per feature level, there are differences in the distributions. And then the third point is more applicable from a practical point of view, because especially in the microbiome, using the bioinformatic tools that we use, for example, Metaflan or Megan, we see that the data types sometimes can be proportion, but they can also be counts if we are possibly talking about amplicon sequencing. And then if some sort of normalization or transformation has been applied, they can also be continuous. So I am not aware of a tool that is actually able to analyze all these three data types in a single framework, which is, for me, is a bit disappointment from a practitioner's point of view. And then there are technological variability both within and between omics, which is again biologically makes sense. I think it should be technological and biological variability. So with these 4 points in mind, this seems like a very difficult problem. But fortunately, even though it looks like a difficult problem, statisticians or some other elegant mathematicians have done the job that we can actually use some of the well-studied distribution for this purpose. So one of the distribution that I'm going to introduce today is the Tweedie distribution, which is actually in the genomics community is somehow overlooked. in general, but it's very popular in some other areas, for example, health insurance applications, because they have also zero inflated data or data with a lot of zeros. So I don't want to go too much details here, but this is a very flexible distribution, and some of the distribution are shown in the right-hand side plot. I just want to emphasize that usually, if you think of a normal distribution, for example, there is a mean parameter and a variance parameter. Similar for a negative binomial distribution, you have a mean and dispersion. But Tweedie is a little bit more flexible because it has one more parameter. So it's a 3-parameter distribution. So apart from the mean and variance, it has an index parameter, which I am denoting as P. This index parameter is not only just an extra parameter, but it's also doing a lot of work to make this distribution flexible. So in the sense that this actually, the index parameter determines what is the mean and variance relationship. And in other words, if I have to simplify further, what is the distribution of a particular feature? How is it more more skewed towards right, or is it more skewed toward left? All this is determined by the index parameter P. So which is really cool thing for a distribution to have. And then another great part about Tweedie distribution is that this index parameter can be estimated in a very efficient manner, like what is the optimal index parameter for my feature. So by optimally mapping that per feature distribution, so you look at the distribution of your data, you determine the best P or the best index, and then you can actually capture a lot of variation that you see. So for example, across environments, is it soil versus human? If I think of other 2-parameter distributions, it won't be able to capture that because it has a fixed mean variance relationship. And that's where I think challenges begin, because if you take a model developed for human microbiome and try to apply to soil, for example, or some other environment, sometimes the results don't make sense, or there are not enough power because of the differences in the environment. And similarly, across features, like depending on whether we're talking about microbes, genes, or pathways, the amount of zero inflation, the distribution does, I think that differs a lot across different features or as well as data types. So again, depending on whether we're talking about amplicon versus metagenomics, I think the zero inflation and other distributional properties vary a lot. So good part is that we have this Tweedie distribution. So on the right-hand side, I'm varying the index parameter, and we see that it can actually capture some of the observed distribution that we see in a real data set. And this is not only index, because there are 3 parameters, we can also actually fix the P parameter or the index parameter, and then possibly vary some of the other parameters like dispersion in this case. By doing that, we can see that we can actually capture some really extreme distributions that are observed in real data. Usually that's not possible for using other 2-parameter families. So that gives the Tweedie distribution a lot of flexibility. Another last part, which is mostly from a statistical point of view, because of this 3-parameter like a generalized form of distributional property, it actually includes many distributions as special cases. For example, P equal to 0, it becomes a normal distribution. P equal to 1, it becomes a Poisson, and so on. So that overall flexibility with the index parameter is the main motivation of using the Tweedie. And that also, if you connect the dots here, that we are really looking for a unified framework, in the sense that the model should be able to learn what is the best distribution for this data and fit a model accordingly in order to have better control of FDR at the end, as well as maintaining good power. I am jumping towards the single cell example because I didn't have negative control and positive control data sets in the microbiome. So on the left, we have verified this hypothesis that this index parameter should be very different across data sets. So what we have done is we have taken some negative control data sets. In this, negative control simply means there is no biological heterogeneity that is expected. But these are single cell data. But as I said, single cell data are usually very similar to microbiome. But what we have seen here, which agrees with the literature and the biology, is that in the negative control data, the index parameter P is fixed at 1. That means it's not doing any sort of we are not seeing any sort of differences in the mean variance relationship or the distribution. But on the other hand, in the green ones, which is the real world scRNA-seq, there is a distribution around how it varies between 1 and 2. So this actually further supports our hypothesis that if we can map this index parameter better on a per feature basis, there is a chance that we can get a power boost. This index parameter can also be thought of as a configuration parameter or a tuning parameter. Again, this is optimized on a per feature basis, and also it's automatic in the sense that the user don't have to do any further tuning. So it will be the best P that it can find based on the data. So that allows the optimal fitting of the per feature properties such as heavy tails or zero inflation across different omics layers. Now I'm going to move into the Tweedieverse methodology and validation. So given that we have the Tweedie distribution that was motivating just from looking at the surface, what are the different characteristics of the Tweedie distribution, we took that Tweedie model and framed it as a generalized linear model. Again, I'm not going to details there, but it's a linear model where you can flexibly model the mean, but using a Tweedie distribution. So usually the GLMs are popular for negative binomial and Poisson and so on. But with the Tweedie also, we can basically take advantage of all the infrastructure that whole GLM family has. That will be clear later on why that is important. But I think, again, going back to our goal, so on the left-hand side, that's our data sets, possibly omics 1 to omics 3, and you can imagine that can be your taxonomic features, pathway features, and for example, metatranscriptomic or metabolite features. You have collected these 3 layers, and then you are on the middle part, which is the black box, is the Tweedie model. And because there is a P parameter, so you are assuming that your Y, which is per feature basis, you are estimating the mean, variance, as well as the index parameter on a per feature basis, and then doing the modeling. What happens is you are not really changing the paradigm when your omics data is changing. That's usually the norm in the regular setup, that, oh, I have metabolites, so I have to switch to a different tool, or I have some other layer, that's why I have to again switch to a different tool, or my experimental design is a bit complicated, so I have to now switch from DSeq2 to some other tool. So that type of interoperability issues is now gone, because you have one single model for all these data types, and then at the end you are getting the inference at all 3 layers. And that also, again, goes back to the point of interconnectedness. Because you have applied the same model, I think these results are much more comparable. For example, if you have applied Wilcoxon or negative binomial to one data type, and then linear model to another data type, and if there is a inconsistency in your results that you expected, I think it's very difficult to say whether it's model specific or it is the biology. But in this case, you have ruled out the possibility of it's a model difference. So that means you can understand or interpret the biology much better, because you have applied the same model, estimated using the same distribution, and everything remains the same. So the expectation of interpretation is much more straightforward. And I'm not going to too much again what are the different properties and advantages of the Tweedie, but to summarize, it's self-adaptive. This is the term that I'm using, and flexible. Self-adaptive because it adapts on its own. I'm not saying that it's an AI, but it's most likely to be like AI, because it's really estimating the P parameter on a per feature basis without looking at any of the metadata, and then fitting the model. And then it handles the zero inflation. I think the complex chart that we saw before, it takes care of that. If you have more zeros or less zeros, zero inflation versus not, that will be taken care of by the P parameter. And it's versatile because, as I said, it's a generalized linear model. So generalized linear model is a very strong set of tools that you can actually really fit some complex models, such as longitudinal or multiple covariates adjustment. All those are actually carried on because of the GLM framework that it belongs to. And then the last part, it's it's agnostic to the data type. And this is really important because most of the computational tools are only built for one type of data. Is it only proportion or only count or only continuous? It's not all of them. So the Tweedie solves that problem to some degree. And as a result, it provides a unified framework. So it's an R package and in a development version available from GitHub. So here is the link. And I'm going to introduce some nomenclature and algorithmic details. So there are two models. One is non-zero inflated, which is called CPLM. That's again, because it's also connected to a compound Poisson linear model. That's a separate or a complicated name for Tweedie, but it's really the Tweedie model, CPLM. And then there is a zero inflated Tweedie, like if your zeros are really more than what you expect, even not being explained by the Tweedie p parameter or the Tweedie distribution, then you might need a further zero inflated Tweedie distribution. Again, it's not a difficult thing to do, but just in case people have, so this is again a multi-model system that we are trying to build, that people have the opportunity to fit multiple models, be it CPLM and ZICP. And also there is an automated version, even though I'm not going to talk about that, it automatically chooses the best model for you so that you know that it fits CPLM when the CPLM is more appropriate, ZICP when ZICP is more appropriate. And remember, within each CPLM and ZICP, it's actually fitting multiple models based on multiple index parameter and choosing the best one. So it's really like a model selection in disguise. It's not showing because it's choosing the P index parameters smoothly, but it's actually trying to find the best P or best distribution first to ensure that your model is possibly the optimal one. Before again going into some of the results that we are going to see for Tweedieverse. So one thing that since this project started when I was a postdoc at Harvard, at least that from that point, or at least before that point, I think one of the things that most of the benchmarking, previous benchmarking missed was the evaluation of false discovery rate. So it was usually ignored as a metric of evaluation. So it felt like it's the elephant in the room that nobody wants to talk about. So people develop a tool, they show that it has great AUC and power without really reporting the FDR. And what happens is somebody else comes up with a different and independent mindset, and from that evaluation it reveals, oh, this tool has some issues with the FDR. The FDR is really high, but it's not. Usually, most of the benchmarking avoided that initially. It was because based on some other types of metrics like AUC, which should not be interpreted actually. It should not be super enthusiastic if you see an AUC of 1 if your FDR is high. Similarly, we based on that, or at least motivated by the fact that no FDR is underreported in the benchmarking that were previously done, we did a similar benchmarking recently in a separate paper, and we also saw the same thing. Again, not the topic of today, but this was one of the motivation of developing Tweedieverse. So one thing that we saw here, this is again a synthetic data evaluation. On the top panel is the sensitivity, on the bottom it's false discovery rate. At the 5% level, most of the method on the top of Maslin 2, that's the method that we developed as a part of this, but this is a biobakery tool as part of the Hut and Har lab. Maslin 2 was the only method that had reasonable power. Again, the power was pretty lower side, but the FDR was well controlled. But most of the other methods that claim to have a lot of power or sensitivity to detect features actually also had very high FDR. So in the sense that you really can't trust those methods for reproducibility purposes, because it finds most of the features as differentially abundant when it's not. So that was our finding as well. So the natural question then was to ask, can we actually achieve the best of both worlds? So in the sense that with this Maslin project, for which there is a preprint available, the link will be in the next slide. We found that a simple linear model, which is what the Maslin 2 is based on, actually can control the FDR, which has been an issue with the existing method. But we also see that the FDR control comes at a price of low power. So the next natural question to ask is, can we achieve the best of both worlds? And to answer that, we did a large-scale comprehensive benchmarking. The idea of the benchmarking is that we know the ground truth, and somehow similar to a spiking experiment where we introduce an effect or introduce some sort of association. I think that's the term, possibly is more close to a statistical association. Then since we know that what are the features that we have spiked in, and we know that what are the metadata, or what are the features that are actually associated with the metadata of interest, in this case, for example, cases and controls, then we can actually do a very comprehensive benchmarking by applying different methods to this data set, where we know the ground truth, because we can easily calculate false positive, false negative, all those different metrics based on the information of this four FP, TP, T, and FN, which are false positive, true negative, and false negative. For this purpose, we use a tool called sparse-dosai. Again, this is a biobakery tool developed in the Huttenhauer lab. So on the left-hand side, you will see that the synthetic abundances, that's actually the most trickiest part. How do you generate synthetic data from a microbiome data without really losing the real-life property? How does it work? It actually works based on a template data set. So that actually, again, makes the tool really, really flexible, because based on the template data set, it tries to optimize certain parameters. Not going into details about that today, but then it tries to mimic the real data and validate using some algorithms, and then outputs a synthetic data set that's very similar to a real data. So what that does is, regardless of whether you have data from human microbiome or other environment, you can actually use this tool to generate synthetic data for your purpose of evaluation, if you want to evaluate a method, or for other purpose, for example, power calculation. So to simplify the narrative here, given the ground truth information, that's what we are trying to do in the replicated synthetic data, that we want to do it many, many times by varying the parameters. We want to see what method has the best average performance over several combinations, and as measured by both false discovery rate and power, not one of them. So which method has the good control of FDR as well as reasonable power. And to break it down a little bit more, as I said, there are 3 steps. I think I'm going to do a little bit quicker here. So on the top panel in the figure left, you'll see that there is a calibration training data. So that's usually your feature by sample metrics. So the sparse-dosai algorithm tries to learn certain parameters based on, I think you can see the details in the preprints. Then it uses those parameters to actually generate, based on some probability distribution, some feature by sample metrics, and there is no effect so far. So that's why it's a null matrix, in the sense that there is no spike in that has been introduced. So on the second step, what we get is a null matrix based on my calibration training data, which can be my any data set from any environment. And then at the last step, we are introducing the spike in a controlled manner, so that we know what are the different features that are being spiked in, so that we can use them as a ground truth information to calculate all those measures that we want to do. Right. So there is a quick demo you can see from the Tweedieverse tutorial. The link is here. You can see it from here, or the description, or also it will be posted in the conference chat. So I think the important point to note, like if you have never used a simulator, you will see that in the first example, you can generate an arbitrary data set with certain features and certain samples and certain effect size. And then you will see that it will also tell you which of the features are true positive. So there is a name attached to it, _TP, to the feature names. And then if they are not, there is no _TP. So that's how actually we used those feature names, like, and the ground truth information to calculate the measures that we wanted to evaluate the methods on. So with that, I will talk about the details of our benchmarking, and these are some of the details that we used in the Maslin 2 preprint that we talked about in the previous slide. And we are actually going to use the same framework for the Tweedieverse validation. So this applies to both Tweedieverse and Maslin 2. So I think you'll see that there is a combination of methods on the first, on the left left top, that more than 100 combination of normalization, zero inflation, regression, and that was really needed for the field because we wanted to be as comprehensive as possible. And then the 10% of the micro features being randomly spiked in, that is again based on possibly biology, that you don't expect more than that, but it's a user defined parameter. You can change that if you want. And then there is an effect size parameter that you can vary as well, like starting from a very small effect size to a very high effect size. You want to see how the different methods perform, and once you have able to done that, you can do this many times. We have done only 100 times. I think that's enough. And based on that 100 data sets for each scenario, you can calculate the average false positive rate, false discovery rate, sensitivity, etc. Again, this whole benchmarking comes in a separate repository, which is going to be listed again in a future slide, as well as in the comment section, as well as in the description. So this is the first set of results that we got from Tweedieverse. So this is a particularly zero inflated microbiome counts being generated from a human microbiome template data. So in the sense that we expect that this data is zero inflated. So that means we need possibly zero inflated model to tackle some of the issues that these methods have. And this is only a preliminary version of the result. But what I want to show here is, obviously, I think the first thing is not surprising that some of the methods, metagenome-seq, GIB, and NECBIN, they actually have inflated false positive rate. I'm showing false positive rate here, but it also equally applies to false discovery rate. Even though if you do not look at the false positive rate, you will be really excited, like it's really high power, right? Because it's close to 1, in some cases more than 0.8. But it's actually not the case when you see the false positive rate. So those are the methods that somebody would not trust. But on the other hand, ZICP, which is the zero inflated version of one of the Tweedie model, has a little bit slightly, I think it's like 0.5 to 0.7, I think, slight inflation of false positive rate, but it has a very good power as compared to some of the existing methods. And this slight inflation is actually not worrisome because we have not done any sort of independent filtering. Usually you can do independent filtering to really track it down to the level that you want. And apart from that, because this is a zero inflated data, CPLM, which is the vanilla or the non-zero inflated version, is actually performing well, even though it has a drop in sensitivity. But it's really, the false positive rate is never above the threshold that we want to control for. But among the methods, if you really want to rank these methods based on the false positive rate controlling behavior, I think, and also highest power or high power, ZICP is actually the best method. And if you obviously allow a small amount of inflation, and then CPLM is the second best method. So that's what we get. So which basically boils down to the fact that we have been able to use this Tweedieverse or the property of the Tweedie distribution to actually achieve the best of both worlds, in the sense that if you compare, for example, with Maslin, I think here is lm.tss, which is one version of Maslin on the left-hand side of CPLM in the right-hand side plot. You'll see that even though it does control the false positive rate, it has low power, which is, again, you do want the false positive rate to go always under the threshold that you want, but also you want high power. So I think this is where CPLM and the ZICP models show improvement by fitting the Tweedie model per feature. So this is the first set of analysis results, but we are extensively testing it on other types of data. For example, the template data set, we are changing that and varying that as well, which will be available in a preprint shortly after the conference. And to further really emphasize that this is a multi-omics tool in a true sense, so we actually took this Tweedieverse to a host single-cell RNA-seq comparison, and there we only took the method that were popular in that field. There also, there is a clear distinction that CPLM and ZICP, which are the blue and purple, have the highest AUC, and it controls the FDR at the 5% level. So ZICP CPLM remain one of the best methods, even in a separate field or a separate discipline in single-cell RNA-seq. I think the goal is to really take it forward because of the Tweedie index parameter that changes according to the per feature distribution. We are seeing that it actually is applicable to many data types, including microbiome and non-microbiome. We do need to see that obviously how it performs in more challenging environments, for example, soil, but we have not done it so far. But we are hopeful that with the index parameter estimation happening at an optimal rate, we might be able to do better in those challenging environment as well. So with that, I am actually going towards the case study using the integrated human microbiome project, which is the part 4 of my presentation. So those of you who are not familiar with integrated human microbiome projects, it's actually the second part of the human microbiome project that was completed in 2012. And this second phase is called integrated HMP or IHMP. So one of the, I think it's an umbrella of studies, but one of the study that was that happened at the Broad Institute was based on the disease area IBD or inflammatory bowel disease. So since there was no study available till date that investigated the microbiome and microbiome multi-omics to see how it relates to IBD. So what are the different properties of different microbiome and other omics data types that are implicated in IBD? So that was the main goal of this study. And this is also one of the largest multi-omics data, if I am not mistaken, especially at least until 2019. So that gave us the opportunity to actually apply Tweedieverse to this data set because it's a rich data set and it also fits the purpose because it's a multi-omics as well. And to go a little bit more details on this, and these are some of the summary statistics based on how many samples available. So there are stool, biopsy, blood, and different data types available. Obviously, this study was optimized for metagenomics. So there are more metagenomic sequences as compared to other data types. I think metabolites come second or third, and there is a metatranscriptomic layer. And also there are a bunch of host site measurements as well. But I think the point here is that this is a multi-omics data set, but there are also challenges that you don't have too many complete pairs. That usually you have pairwise data available for many points, but not complete pairs. But I think that's even though that's the case, I think we can use this data set because we are not doing any paired analysis, but just for our information, we know that if we want to connect the dots for two layers, and also across multiple layers, we may not have the same set of sample size across comparisons. But again, since this is a rich data set, we wanted to test whether we can actually recapitulate some of the findings we had in the original paper. So these are some of the features that are implicated, and good part is this figure is being generated by Tweedieverse because it's part of the visualization that is embedded in Tweedieverse itself. I'm not going into too much again what are the features that we have selected, but one thing to note is that the overarching theme of the Nature paper or the IHMP study was that which is again possibly a known knowledge, especially, but it was not verified in a large multi-omics cohort, which is large population cohort, I would say, using multi-omics. I think the overarching theme of the paper was that there was an increase in facultative anaerobes like E. coli at the expense of some more commensals or SCFA or short chain fatty acid producers, for example, Faecalibacterium prosnitzi. So these are some of the plots that agrees with the definition. So on the x-axis is a dysbiosis, which is a measure of disease for the IBD. So for the commensals, you would expect that it goes down. So there is Faecalibacterium prosnitzi, and also there is a another novel species that was identified in that paper in the middle, and then E. coli, which is the opposite because of the reverse connection. So the point that here that makes Tweedieverse really interesting is that the original paper IHMP study is a longitudinal study, so it analyzes the whole data set and finds this finding. But if you restrict yourself to the baseline samples only for this data set, what we saw that the original study actually could not find any differentially abundant features if you look at the baseline samples only, even though the study or the paper claimed that most of the IBD versus non-IBD, they are usually patients with some problems in their gastrointestinal tracked, which means that they are more or less similar in nature. So there is a chance that at the baseline, when they were recruited, there should not be any differentially abundant features. But when I was analyzing the data as the statistician of this study, I realized that that was possibly an argument, but then another argument was that the model that was being applied to test it was not powerful enough. So that was my intuition, that if I take the baseline samples only and do a testing, and the original study did not find any significant difference, but which to me didn't make sense because it's an IBD cohort. I do expect the IBD to be different from non-IBD. I applied Tweedieverse, that's both the CPLM and ZICP models, to the baseline samples. So what we saw that some of the findings that were in the original study based on longitudinal data, that means a very large data set, whatever finding that was, we're actually able to find it using only the baseline samples, which is only less than 100 or so subjects. And Tweedieverse CPLM actually finds 15 such features, and Tweedieverse ZICP finds 28 such features. And those include some of these findings that you can see from the scatter plot. So to summarize, it recapitulates original findings, but based on less data. So based on only the baseline profiles that was not found by the original study. So that means there is a chance that Tweedieverse has increased power as compared to some of the popular methods. And that brings me to the last point, last part, and this is the part I'm really excited about. So far we have talked about how to unify different methods and what is the best way to get there using the Tweedieverse. And Tweedieverse seemed to solve some of the problems that exist in the current literature. But I really want to make sure that as a conference attendee, you really get to use this tool, even though you don't work in the human microbiome. So I want to make sure that we connect the dot here just one more time, like what we're trying to do and what is the issue, and then how we are actually solving the problem. So this is a video, I'm sure that many people have seen it. This is a snapshot from that video, biologist talking to statistician. So before Tweedieverse, I think the statistician will give you this complex workflow. If you go and say, I have a microbiome multi-omics data, can you tell me how to normalize and how to do differential analysis and how to adjust for multiple covariates, etc. And then this will be the answer of the statistician. No matter how much you argue. But what after Tweedieverse it is going to happen is that it becomes a very simple set of decision rules. So regardless, if your questions are complicated, like what if my input data is count, proportional, or continuous, the solution is simple: use Tweedieverse. And if you are not sure whether to use a zero inflated versus non-zero inflated, again, Tweedieverse is one of the tools that lets you use different versions of zero inflated and non-zero inflated models. You want to ensure FDR control and power, use Tweedieverse. And if you have repeated measures and multiple covariates, again, use Tweedieverse. I think only part that I can think of where Tweedieverse may not be recommended as of yet based on our validation. What if you don't trust the validation that we did using sparse DOSA or the human microbiome template? In that case, I think, again, the solution is actually simpler. Use Tweedieverse. But if you are unsure that the validation that has been done is going to replicate to your own data set, there is a very simple way or easy way to just use sparse DOSA, because there is tutorial how to generate new data set, new synthetic data set using sparse DOSA, and then run a very simple evaluation using Tweedieverse or any another simulator based on your data set. So that would be still, I will say that the complex workflow that we had has made life much more easier because of the flexibility of the Tweedie distribution. And I must say that Tweedieverse is currently the only tool, to the best of my knowledge, that ticks all the above boxes. I think especially the first one is really important. Most of the tools are only either count-based or proportion-based or continuous, and they are not multi-model. That means if a tool has decided that negative binomial is going to be my underlying model, as an user, you are actually not going to change that. That's hard-coded. That is how most of the tools are developed. But here, if you have the opportunity to change the model if you want, there are 2 models currently, but the goal is to have multiple models for the users. So with that, actually, Tweedieverse is really the only tool, especially when it comes to microbiome multi-omics differential analysis. And if you want to get started, the good news is that there is a tutorial and benchmarking repos separately for the purpose of doing either evaluation or using Tweedieverse. So the links are here, and links will be shared again with the attendees in the comment section, or as well as in the description. So I think just to summarize, there are method specific or tool specific GitHub repo. So you can see that Tweedieverse is one of them. The first link is that, and also Sparse-Dosa, there is a second version as well, which are the simulators or the benchmarking. If you want to do benchmarking, that would be the tool to go. So those have their own repo, but in order to actually run this whole, I would say the whole machinery of generating the data and then generating the results, that is actually in the another repo called Benchmark Microbiome, which is the second link. And then the associated package that we talked about, Maslin 2, is also has its own repo that's in the link 5. And the good part is support is available. If it is a biobakery tool, there is a biobakery forum from the Hartenhauer lab. If not, if it is possibly in my personal repo, then you can ask any question through the GitHub issues. And these are the preprints that are related to this talk: Sparse-Dosa, Maslin 2, and eventually all of them came in 2021. And the main microbiome specific Tweedieverse preprint, which is currently we are preparing, is the last one that is going to come up soon, which is Tweedieverse, a unified statistical framework for differential analysis of multi-omics data. So that will be available very soon after the conference. And finally, if I just want to conclude the main take-home messages, we have developed a flexible and self-adaptive framework for multi-omics differential analysis. And this is motivated by the fact that there is no such tool currently that exists. How we have been able to do that is really not any sort of magic happening. It's really a distribution that exists, which is a well-studied distribution, has been there for many years, but not used by genomics or computational biologists for some reason. But that using the property of this distribution, which tries to optimally map the per feature distribution, it actually is able to beat some of the existing methods with respect to FDR control and power. Again, I don't expect Tweedieverse to be like the best method in every scenario, but I think because of the flexibility of the per feature, it's going to be a more or less robust method, sometimes maybe performing a little bit worse as compared to some of the methods, but more or less, I think I give the example of random forest in machine learning. It's not always the winner, but it's a more or less reliable method in the machine learning field. So I think I expect Tweedieverse to be a very similar method, even though it may not outperform all the time. So it resolves the long-standing interpretability. I think that's the part that we got motivated to do this, that whenever a new omics layer or technology comes up, there is usually a better way to use the existing method or existing tools. But for some reason, we are not doing a good job in resolving this long-standing interoperability issue, possibly because we didn't identify flexible distributions enough, or most of the popular distributions that are being used are possibly not flexible enough. With the Tweedie's flexibility, I think we can at least address some of the interoperability issues across omics and within omics. Again, this is important because even within microbiome, you can see there is a lot of variation in the per feature distributions, and across omics it's even more diverse, and this also applies to environments and whatnot. And then as I said, this is a multi-omics tool, so validation in other omics modalities is currently ongoing, and some longitudinal extension as well as automated model selection, as I little bit mentioned in one of the slides, is also ongoing. Our package with in-depth tutorial available. The tutorial is being upgraded all the time. All the links will be shared with the attendees either in the description as well as in the comment section. And obviously, feel free to reach out to me either during the question answer session or later on if you have any questions. So with that, I would like to thank all the co-authors and the collaborators of this paper. And so there are, the names are here. The reason I wanted to mention Huttenhor Lab, which is my postdoc lab, because this idea actually was conceived as a Harvard postdoc when I was doing another project that I mentioned, Maslin 2, and I saw that my best performing method has low power, and that was the main motivation behind actually developing something more elegant. And it was just finding the Tweedie distribution that was the starting point that happened during the Harvard postdoctoral fellowship. I didn't have time then to implement it, but as a mark scientist, I was able to do it. So with that, I would like to take questions, comments, suggestions during the Q&A session. I hope you find this tool useful, and especially for the purpose of analyzing your own data.
Great. That was perfect. I'm going to stop recording.